As indicated earlier, now we will discuss the role of mass spectrometry in analyzing the protein complexes. Now, protein molecules are very important in discussing the role of molecular biology and in the study of molecular biology. So, mass spectrophotometry is, of course, employed for the detection and identification of the protein molecules. And mass spectrophotometry is used to identify the partner proteins, that is, the proteins which interact with particular protein of interest. You see, in an organ or in a cell, the different proteins, they interact with different proteins, and with the help of mass spectrometry, we can use to identify the partner proteins or the proteins which accomplish or which join or which work with the other particular protein in the cell. Now, interacting proteins, they can be isolated by a number of methods. That there are methods where we can isolate the interacting proteins, and the method includes: one is immunoprecipitation of tagged protein from cell transfection. That is, by the immunoprecipitation of those proteins from cell. Another method is affinity chromatography, where we can separate the different proteins. And then there is a surface plasmon resonance, that is SPR. Now, these all methods are very important to separate or to identify the interacting protein. Among these, surface plasmon resonance SPR technology is very, very useful and has widespread application for biomolecular interaction analysis. That is, when the proteins, they interact with each other, the surface plasmon resonance, it has application and it is used to find out that which proteins are working over there. Now, SPR is also applicable during characterization of protein ligand and protein-protein interaction. You see, in the cell, what happens that the proteins, they will interact with the proteins, but at the same time, they can interact with certain ligands, chemical ligands, and SPR is used to detect or characterize those proteins which are interacting. Now, direct analysis of protein complexes by mass spectrometry is also possible, whereas it is, as previously said, it can be by the plasmon resonance method as well, but direct analysis can also be done by mass spectrometry. Accurate molecular weight of large biopolymer, such as proteins of mass which are greater than 400 kilodalton, that is the intact virus particles of molecular weight of 40 into 10 raised to power 6 or 40 million delta units that have been analyzed using ESI and TUF techniques. Similarly, many other protein complexes have been analyzed by mass spectrometric techniques, which are very useful to render the analysis and the characterization of these materials. Now, icosahedral virus, because which is consisting of single-stranded RNA surrounded by a homogeneous protein shell with a total mass of 6.5 into 10 raised to power 6, that is 6.5 million Dalton, that can also be detected, and that can be detected by the mass spectrometric technique. So you see that in this case, we can use the mass spectrometry technique for detection of molecular weight of RNA as well as the RNA of different sizes. Similarly, the proteins and interacting proteins which are in the cell. And this technique is extremely useful and give and handle to the molecular biology to study these two important molecules.